Thanks to everyone who's answered the poll. And just out of interest, great to see that we've even got an overseas participant today and many regional participants as well watching, and very varied audience in terms of lung transplant recipients, healthcare professionals, supporters, etc. So many thanks. So back to you, Bron, to introduce our final three speakers today.
Okay, thanks very much, Catherine. So moving on in the program, we've got another three speakers coming up. I'm just going to introduce each one of them and their topics, and then we'll hand over to start the presentation. First of all, there's Stephen Hiho, who's our PhD candidate supported by Lungitude. He's completed a Bachelor of Science with honours in genetics, and he actually works at our Victorian Transplantation and Immunogenetics Service, or VITAS as we call it, at Red Cross, where he does a lot of tissue typing and cross-matching of donors and potential recipients. And his PhD is about assessing new tools for improving donor and recipient matching. And he's going to present to us today better matching of donors for lung transplant patients. Moving on to our next speaker is Dr. Lucy Sullivan, who also is a very smart lady. She's completed a Bachelor of Science and PhD at the University of Adelaide, and she's actually an immunologist, understands things that many of us would never understand about the immune system. Her early research focused on lung physiology, and that's where she developed her interest in immunity and the immune system. And she has been involved with our program for quite a few years now, has won many awards. And she's going to present to us, should we always be matching our donors and recipients? So that's going to be an interesting talk following on from Stephen's talk. And then our final speaker today is Dr. Louise Fuller, who is our senior clinical physiotherapist at the Alfred with Transplant Services. She's also an adjunct associate professor at the physiotherapy school at Swinburne, and she recently completed her PhD and was in 2019 was awarded a Churchill Fellowship to study rehabilitation models overseas. Louise was, along with Christy Elmsley, senior dietitian in our department, was the first winner of the Jeff Gittes Lung Transplant Fellowship in 2019, and so she's going to give us a bit of an overview of what that project involves. So I'm going to hand over to Steve now to start his presentation.
Well, thank you for having me here to have a talk about what I do and the research we are undertaking. I'm going to try to talk about what we're doing to identify better matched donors for lung transplants and what we can use, tools we can use to do this. So just a quick recap on a few new things that have occurred at the Red Cross. We are now no longer the Red Cross Blood Service, and we are now Lifeblood. So this is a name change. Basically, they say it sort of incorporates more of what the products we provide and the services we provide. And the second, but probably more major change in terms of transplantation is out of the National Organ Matching System program that we used to use since 1999. Became outdated. It wasn't keeping up with the technology that we needed. And so we've changed to Organ Match. After a few teething problems early on, I think we're running at a good speed now with this. It's a new system. It's compatible with technology that it is at the moment, but also has the room to help accommodate further improvements in technology, which very much will help us when we find get a better understanding of what we can use with HLA. So give a recap about VITAS. It's a tissue typing lab. We do the HLA tissue typing for transplants, such as lung, heart, liver, pancreas, and intestine. We do a lot of testing for bone marrow matching and also for platelet support. We match for all the bone marrow donors. We have to do the HLA typing for those and for the core blood bank. But we also do some gene testing, such as celiac and narcolepsy, and some autoimmune diseases. And of course, the on-call part is the 24/7 role that we play, and that's in the deceased donor process. And determining which patients to do transplants with. Quick recap about this process. So once we get a heads up of a donor coming, we get the donor blood from donate live, and we extract the DNA and do a HLA typing. So the HLA typing the level of HLA typing we can do now has improved greatly over the last few years. This is a quick turnaround typing. This stage is a 2-hour process. So we do that on the donor. Now, all patients are HLA typed on waitlist when they're waitlisted. So we already have that information in the system. And after we do the HLA typing, we go to the cross-match. So the cross-match is using the donor cells that we extract from the blood and matching it and cross-matching it with the patient's serum. So basically, this gives us an understanding of how a transplant will react. This is where we get some people may have heard the cross-match positive result. We don't want a positive result. It means the rejection is more likely. And after we get the cross-match, we do the DSA calculations, which are the donor-specific antibodies. This is the thing that's always talked about. We have to sit down and use the HLA typing of the donor and look at any antibodies that are identified in any patient that's compatible nationally for kidneys and for every other organ as well. And we determine if the antibody that the patient has is directed towards this donor. Sometimes that can be quite a complicated process, but nevertheless we do it. And then we issue these reports. So these go out to the clinicians and the coordinators and say, but basically, is it CDZ positive, and gives a rough estimate of the risk involved. What is HLA? HLA is a very highly polymorphic surface protein. So polymorphic means there's lots of variations of it. There's thousands and thousands of it. So the more variations the HLA show, it's better to regulate the immune system. It has the ability to see other things and stimulate antibody and clear viruses. So there's two main classes, class 1, aptly named, and class 2. Class 1 is the T cell HLA. It's codes for the HLA A, B, and C, and class 2, which is the DR, DQ, and DP. Both have very important roles in transplantation. Class 1, we do a lot with platelet support because platelets themselves don't have class 2. But we're finding lately that class 2 is more important in the transplant role. So why is HLA important in transplantation? So we have a bit of a look here at what areas of the body express HLA. And we could see the highest expression of HLA, the bone marrow right down the bottom. So the bone marrow, we do HLA matching, and probably everyone is aware that you need a perfect match for bone marrow to get a successful outcome. That's what we're talking about. We're talking about HLA matching 10 out of 10, 12 out of 12 level. And then we have the kidney, which we've been doing HLA matching in kidney for over 30 years in Australia. And they've got a moderate expression of HLA, but right in between those is a very high expression of HLA at the lungs. So no HLA matching is done anywhere globally. All we do is that DSA assessment. That's anywhere in the world. And for reasons I'll explain later on. So the HLA in the kidney transplantation, this is the graph that gets updated every few years. So as I said, in Australia alone, it's been over 30 years. So the better matching, the better chance of survival. So when we're looking at this graph here, we're looking at 0 mismatches. That is a perfect match. And then as we increase the level of mismatching, we increase the risk of, we decrease the risk for graft survival. This is for just a few of the HLA genes, and I think further work has tried to demonstrate this into other HLA genes, but at the moment it's A, B, and BR. So if you're a good match, you do very well. So what about in the lungs? How has this worked out in the lungs? So looking at the A, B, and BR, it looks similar in this study done in 2010, where you get a mismatch. If you avoid a mismatch, you do much better. However, if you look at A, B, DR as a combination, and the way it works, it doesn't appear that way. You can have a 0 mismatch performing pretty poorly, and sort of equivalent to a 4 to 5 mismatch. So this study, other studies have been done, giving similar results, whereas it's almost as a random effect. So a perfect match doesn't really equate to better long-term survivals. So why is that? So we have a look at the HLA matching. This is a very common HLA allele, so to speak. It's a DQ3, or as we used to say, DQ7. The majority of the people watching this right now will express this DQ7. It's a very common allele, and that's the genetic sequence that codes for this. So if we're going to talk about allele matching, we'll have to match all these genetic bases from the donor to the patient. That is just for one of the alleles, and everyone has up to 6 genes, 2 alleles each. That's a lot of matching. Should we be allele matching? So, and if so, what do we match on? A, B, C, D, R, D, Q, or D? And that's very unlikely. We can't do this. It's very hard to do in bone marrow transplants. We can't find it in kidney transplants. So, getting a well-matched lung at that time isn't worth it. So, if we look at HLA alleles, obviously there's ever-expanding a list of alleles that we've come across. There's over 30,000 at the moment. By the end of the year, that will increase even more. This is because a lot more labs are doing a lot better testing. We're discovering in our lab 1 new allele every 2 weeks. And so matching for all these is nigh impossible. Australia, as it is, has one of the most diverse HLA populations in the world. We've got Aboriginals to Islanders to the Middle East to Asians to every other combination thereof. And because of that, the combinations of HLA are very rarely seen elsewhere. I myself have only 2 matches in the world for bone marrow matching, and that's from 50 million donors. My HLA isn't unique, but the combination of them is obviously a bit harder to find. So, this is why we're looking at HLA eplets. As I said before, this is the genetic sequence of the DQ7 or DQ3, and it encodes for the whole HLA protein, as you see there. However, what we have been found recently is it's not the whole HLA protein that is causing most of the damage. It's just the top part, binding cleft. So this is the part that the immune system recognises. And from this area, we can narrow it down to a few hotspots in the sequence that we need to look at. These hotspots are eplets, and these are the regions that are exposed. So the way that the HLA structure is twisted and turned, it's not always in the right order when it comes to the cell structure. However, if we can just match to these known hotspots, then we have a better chance of finding a really good match, and we can almost sort of avoid the rest of the HLA genetic sequence. So what our work has looked at are these HLA eplets. Can they be used to predict outcomes in lung transplantation? And what level are we allowed HLA mismatching of these eplets could still have outcomes? So it's not really a HLA matching as the title of this talk presented. It's more of a compatibility. We know there's going to be mismatches. What level of mismatches are we able to do and still have a very good outcome? So what our findings have shown is very similar to others at the 5-year mark. We had a really good outcome for low-level mismatching at 5 years. However, for the overall patient survival, this improves. So the better the compatibility, the better the outcome. So this is still early stages, and we're hoping to really get these implemented at Red Cross. But nowhere else in the world is discovering this, using this at the moment. And because of the changes at Red Cross, we're able to possibly implement this sooner rather than later. In summary, what we're looking at is compatibility rather than matching. It does improve outcomes, as we can see. It's not currently performed. However, we're hoping to sort of implement this into that pre-transplant assessment that we do in the donor process to give a better idea. And that's it. Thank you very much for having me on, and thank you to Lungitude for supporting the work.
Thanks very much. Unfortunately, your sound was a little bit in and out then, so but we'll leave any questions and we'll move on to Lucy, who's going to do her talk next. Thanks, Lucy.
So Steve has introduced HLA, which is great. And I'm going to be talking about a bit of a spin on this around, should we always be matching donors and recipients? And I could have actually also called this talk When Science Throws You a Curveball. But I should say that this is pilot data that has been included as part of an NH and MRC grant. And as Glenn mentioned, this is where Lungitude is really important in providing us the support for pilot studies that we can use to leverage bigger funding sources. So thank you to Lungitude for their ongoing support for not only this project, but lots of other work that we're working on as well. So this is what the textbook says. The textbook says that transplant rejection is caused by the recipient's immune system. And the recipient's immune system responds to these foreign proteins called HLA that Steve's talked about. And he's also talked about the fact that HLAs are very different between different people. So when we talk about HLA in the transplant world, normally we're talking about HLA-A, HLA-B, and HLA-DR. And basically, you inherit these sort of sets from your father and your mother. So if the letters and numbers aren't that important, but just note they're A, Bs, and DRs, and they're 1s, 2s, so they're different. So all of the HLAs are different, the Bs are different, and the DRs are different. So the possible outcomes from this particular mother-father combination is a red and a blue, a yellow and a blue, and a red and a purple, and so on. So you sort of inherit these sets of genes from your parents. And how this works in the transplant world, your lung is covered with these HLA proteins, and these are essentially your proteins. They are extremely variable from person to person. And these molecules is what essentially leads to CLAD. Because when we have a donor lung that has different HLA, we can get a recipient T-cell response, or we can get antibodies that bind to those HLA, which ultimately lead to this phenomenon called chronic lung allograft dysfunction, or CLAD, which is essentially chronic rejection. So this is what the textbook says, as I've mentioned, better HLA matching equals better transplant outcome. I'm not saying that's wrong, that's absolutely right, but it's when we're looking at HLA-A, HLA-B, and HLA-DR. And because I've just told you we've got 2 copies of each, we end up with this figure essentially exactly the same as what Steve's shown you, whereas if you have more HLA mismatches, you have a worse transplant outcome. So in real life, we have lots of these different HLA molecules that are encoded in this chromosome. You don't need to remember them all, but just know these are all the class 2 molecules that Steve talked about. And my research is largely focused on these class 1 molecules, where we have A and B that we've already talked about. There's also these other ones. In particular, I'm going to be talking to you today about this HLA-C. So poor old HLA-C is a bit of the poor cousin of the HLAs. So HLA-A and HLA-B are always looked at, but what about HLA-C? So what about HLA-C? Just very, very briefly, there are basically 2 flavors of HLA-C, and we're going to be calling them HLA-C1 and HLA-C2. And because I've told you that you get 1 set from each of your parents, these are the combinations that you can be. You can be a C1-C1, you can be a C1-C2, or you can be a C2-C2. So are there any differences in transplant outcomes in recipients with different HLA-C? Yes, there are. So this is data from Steve's cohort that he's shown you. And we're looking here, so freedom from rejection. So we want to be up here. We want to be, this is 100%. We want to be free from rejection. And this is over time. So as you can see, over time, we get rejection. But recipients that have 2 copies of HLA-C2 do significantly better than the other combinations. And what about if we add in the donor HLA-C onto this? Well, this is where it gets like really crazy, because these are only now looking at this improved HLA-C2 recipients. And this curve out here, which is the recipients that do the best, are actually the ones that get a completely mismatched lung, an HLA-C1 lung. So this was a big surprise when we saw this data. And it was sort of a bit of a lesson as well in that science is cool and interesting, and you need to look at the data from all sorts of different angles to get to the right answer. But you're going to say that doesn't make sense. Or what is going on? And to be honest, I don't have an answer for you because it is a bit of a curveball. But perhaps we can explore some reasons as to why this might be. So what we know about these molecules that we always discuss, HLA-A and B, is that these are seen by T cells, whereas HLA-C is more commonly known for being seen by this other cell type that have a really cool name called natural killer cells. So what are natural killer cells? Well, they're quite variable. They're 1 to 10% of your white blood cells, and they're known for killing virus-infected or cancer cells. And they can also rapidly kill cells that are coated with antibodies. And this is important in the context of lung transplantation because we can get donor-specific antibodies on the lung. So unlike T cells, these guys respond very, very fast without needing to be activated before they can kill cells. But natural killer cells are inhibited by this HLA-C molecule. And we know from our other research that natural killer cells differ in their ability to be inhibited by C1 and C2 molecules. So the question that we're proposing for our NH&MRC grant is, could NK cells be responsible for the different outcomes in recipients with different HLA-C? Now, I don't have the answer for you. I'm hoping that we will if this is funded. But the sorts of questions that we want to ask is, are there different proportions of NK cell subsets in HLA-C2 recipients? And I haven't introduced you to this today in the interest of time, but natural killer cells aren't all the same. There are lots of different flavors of natural killer cells as well. So is the function of NK cells different in HLA-C2 recipients? And can a mismatched lung actually change the function or the proportion of the different flavors of NK cells? So the significance of this research, so as Bron mentioned, I'm a scientist, so this is super awesome for me because we're challenging the existing doctrines and challenging what they say in the textbooks. So we can work towards an outcome. Obviously, it's not just science. What our ultimate goal is to improve survival following lung transplantation. So how could we work towards this in this project? Well, we can look at targeting HLA-C matching or mismatching, as the case may be. I think I may have a little bit of trouble trying to convince my clinical fellows that this is a good idea at this stage, but time will tell. And from a scientific perspective, it's possible that a foreign lung can actually sort of retune your white blood cells into either different functions or different proportions. And if we monitor these over time, we can potentially have a more tailored approach to treatment options following lung transplantation. And moreover, this research is completely novel and it can have applicability in other settings, such as in cancer immunotherapy, where we know that HLA molecules are expressed sort of a bit strangely. They can start upregulating things that they didn't express before. So it's potential for other clinical settings as well. So these are the people who work with me on these projects, as well as a number of other projects that we've got running at the moment, thanks to the very generous support of the Lungitude Foundation. And I also have to have a big shout out to very generous support of our lung transplant patients who donate both their time and their clinical samples, without which this research would just never be able to be undertaken. So thank you very much for that. And just to end this on, this is exactly what we're dealing with in science. So simple but wrong. Most people fall off the edge of the cliff, and this is what we're working towards, complex but right. So thank you.
Great. Thank you so much, Lucy, for helping explain something even more complex in a simple way. And last but not least, of course, we'll move on to Louise Fuller, and Lou's going to talk about her Jeff Gittes Fellowship project which I'll let her explain to you. Over to you, Lou.
Thank you very much to the Lungitude Foundation for sponsoring Christy and my study. And we'll be looking at body composition and muscle morphology after lung transplantation with the interesting name, we've called it Ultra. Oh, the slides aren't moving on. Here we go. Now, the majority of lung transplant patients we know have impairments in skeletal muscle mass and strength, both before and after lung transplant. And this is called sarcopenia, which is defined as decreased muscle mass with at least one of low peripheral muscle strength or a change in their function. So the common methods to assess sarcopenia are using a BIA machine, having a CT scan, or having an MRI scan. And the common methods to assess muscle strength are handheld dynamometry, isokinetic dynamometry of both the upper and lower limbs. However, the quadriceps muscle in your lower limbs is the one that seems to be most commonly tested and reported in the literature. However, there is no consensus in the research literature about which of these tools was better to use either to measure muscle mass or strength. We also know that there are deficiencies in the quadriceps muscle strength and also in physical performance are very common in lung transplant candidates. The exact nature of the reversal of these deficits post-lung transplant is really largely unknown. In addition, there's alterations to the muscle morphology, so how the muscle actually looks post-lung transplant, which is complicated by taking different medications and especially the corticosteroids and in conjunction with tacrolimus. We also know that there are functional changes during and after our rehab program, in particular looking at changes in your 6-minute walk test distance, and this has been shown to improve after transplant. But we don't know what the exact changes in the muscle morphology and your body composition really are. I think some of the recipients who are listening to this talk today would realise that weight gain is really common after lung transplant, and obesity is an independent risk factor for primary graft dysfunction. But what we want to find out from this study is this weight gain muscle? Is it fat, or is it an increase in fluid? So the BIA, or the bioelectrical impedance analysis, has been shown to be superior to just doing measurements for body mass index alone. It's non-invasive, it's very reliable, and it is available in our clinic. We have a BIA machine that most of you may have been put on as part of your assessment for lung transplant. And what it does is it measures fat mass, fat-free mass, skeletal muscle mass, and total body water. And at the moment, it's only used in our screening for candidacy, but it's not used to measure what your actual body composition is after the lung transplant. The other tool that we're looking at is two-dimensional ultrasound screening, and we know that this is painless, it's not invasive, it's radiation-free, it's relatively inexpensive, it's extremely reliable, valid, and it's available. The physio department and the respiratory medicine department have several portable two-dimensional ultrasound machines available. It will tell us a lot of information about muscle size and also that morphology, the quality of the muscle. And in ultrasound world, it's described as the echogenicity of the muscle. It's been used in the ICU setting, relating muscle mass to mobility in ICU scores, and cross-sectional ultrasound has been used in other studies in the past to measure muscle thickness and in particular the size of the quadriceps muscle. So that includes all parts of the quadriceps muscle, the rectus femoris, vastus intermedius, vastus lateralis, and this has been shown to quantify muscle weakness on ECMO patients in intensive care who can't move around. So the aim of this single centre study was to investigate the changes in body composition, including unintentional weight gain and muscle composition, in the first 3 months after lung transplant. And what we're hypothesising is that there'll be an increase in lean muscle mass and a decrease in fat mass. So the overall hope after this research project is that the research will assist in improved exercise prescription and nutritional guidelines for everyone in those first few months after transplant. What we're going to do is we're going to include everybody who's had a single or a double lung transplant, who is an adult. We will be excluding interstate patients because it's limiting in how we can do their baseline measures, and anyone who ends up going to an inpatient rehab facility afterwards with complex medical problems. This is what the ultrasound looks like. So we're going to look at the cross-section and circumference measures of the whole of the quadriceps muscle. And then we're also going to measure the morphology of the muscle tissue on a program called ImageJ. So we're looking at the grayscale measurements. It's a software program. And these are the kind of images that we'll be getting back from ultrasounding the quadriceps muscle in 2 different ways on the thigh. So looking at the cross-sectional mass. Christy's going to be using the BIA. In addition, she's going to be looking at height and body weight to calculate BMI, and then all those other measures that are up on the screen, so that we can get a good indication of what actually your body composition changes are over time. We'll also look at a functional measure, which is a 6-minute walk test, and we're going to be doing some strength testing on the quadriceps muscle using a strain gauge chair, and of course having all the demographic details of your transplant up there. These objective measures will begin at baseline, so when people are listed for transplant, on entry to the rehab program, and again at 8 weeks post-lung transplant. And we've selected just a convenient sample size of 60 people. We think it will take about 14 months to recruit and transplant and rehabilitate 16 patients and follow them up. It just depends on the transplant numbers as they come through. So we're just waiting for some ethics approval. The trial's been registered with the Australian and New Zealand Clinical Trials Registry. And of course, we are so grateful that it's been funded by the Jeff Geddes Fellowship through Lungitude. So both Christy and I are very honoured with that it was the first one. Unfortunately, this project was due to start at the beginning of this year, around March, and there have been major changes to our rehab group format as a response to the COVID pandemic. So it's resulted in a big delay. But having said that, we are hopefully in a good position to start at the beginning of next year and get a clear run through. So I would just like to thank Lungitude again and the whole physio team, Jess, Beck, Mon, Ben, Meg, and all my gym staff for all their support in doing this, and especially to Christy, who can't be on speaking today. So thank you very much for listening, everyone.
Great. Thank you so much, Lou, for your talk. And I just thought this was a good segue into just talking a little bit about the Jeff Geddes Fellowship. The inaugural Jeff Geddes Fellowship, funded by the Geddes family in memory of Jeff, was first started last year. And as Lou said, she and Christy were the first winning recipients of this. Some of you might remember Jeff. You may have met him. He had 2 lung transplants with our team, and unfortunately, passed away about a year or so ago. And he and his wife were very instrumental in helping Lungitude Foundation really flourish to the organisation it is today. And so we're actually going to be announcing or opening up the 2020 Jeff Geddes Fellowship in November. and the information about that will be on the Lungitude website for a small project grants, and so all the information will be available there, and hopefully we won't have quite the impact that COVID's had moving forward for this fellowship to be able to be conducted next year as well. So keep your eye out for that, and if you're interested in applying, go onto the website. It should be up there in the next few weeks. So I just been looking at all the questions online, and it looks like our have been answering all of the questions as they've been going. So unless anyone has got anything pressing that they would like to ask on the question chat line now, I think I might hand back over to Catherine.
Great, thanks so much, Bron. And oh, what a fantastic session we've had today. Jam-packed full of really interesting science and updates of what the great researchers have been doing. So a very, very big thanks. I know there were a lot of questions through the session which were answered on the chat, but as Wendy has mentioned previously or shared previously, we will be sharing the questions and answers separate to this session as well. So I'm very conscious that we've got 3 minutes to close the session, and really want to respect people's time and finish exactly at 2. So firstly, let me just a couple of news announcements, I guess, in relation to a couple of things coming up and to watch out for from your side before I close with thanks to those who have been part of today. So as you know, every year we're involved in the Melbourne Marathon. And despite COVID, this is going ahead in a virtual fashion this year for people being able to complete their marathon on their own at some stage between the 5th and 13th of December. So we'll still be involved in that. So watch out for that and keep that date in your diary. And another note as well is that the impact report, which I know many of you will be waiting for and really enjoy reading, which summarises a lot of the key statistics and further information regarding transplant work, is being shared very, very soon. I think it might be tomorrow, in fact. So just some little reminders for you. Just a big thanks again to our tremendous supporters and our sponsors, without whom, of course, none of this work could take place. So very, very big thanks to everyone. I'm not sure, Gordon, I think we were going to do a check handover, so I'm not sure if you want to do that now before I go to final thanks to our presenters today.
We can do a very, very quick one. So we are pleased, hopefully everyone can see that, to hand over today a check for, and it's a big check, over to Greg. There you go, Greg. It's over to you. So it's a check for.
Okay, yes.
Amazing technology.
Hey, how good is that? So it's a check for $150,000, which thank you, and we have to say a big thank you to all our supporters. And as all the as everyone said from the Alfred, that's to cover the numerous different research projects. I also believe that it's for a machine as well that we bought for Lucy and the team, which is being loaned out to some important COVID research as well. But for me, I think what I've taken away from today is that a lot of the research, a lot of the big projects, and the big funding from the HRC can't happen without the Lungitude support and without all the support from both our corporate supporters and individuals as well. So thank you for that, and I'll hand it over to you, Catherine, to finish off.
Great, thank you, and great technology, seeing that check handed seamlessly across the ether there. So very well done, very well done. So a big thanks today. I think it's been a fantastic session today. Big thanks to our presenters for their great presentations, but more importantly for the work that's underpinning the presentations that they shared with us today. They're relentless in their pursuit of better outcomes for patients, recipients of lung transplants, and support for carers. And we're very, very grateful for that tremendous work that they continue to do and their passion in this area. A big thanks also to Bron and Wendy for really bringing today together for the work behind the scenes to make sure that this all worked well, and of course for the board who support all of the work that's behind what Lungitude does. It's been great to be united today. All of us are united, I think, by our desire to improve the outcomes for lung transplant recipients and their quality of life. So great to have this opportunity to bring everyone together despite the challenges of COVID. So a big thanks there as well. And I think with that, we can really close the session today. Just a reminder that there will be a questionnaire or survey that's sent to you, a very brief one, I promise, and that there will be one lucky attendee from today who will receive a Prezi gift card, which, as we've said before, was donated by one of our very generous supporters anonymously. So a small amount, but not massive amount, but a big thanks to all of you for attending, especially our person who's attended from overseas. So thanks again. Thanks, Bron. Thanks all of our presenters. And we really look forward to seeing you in the future at our next event, hopefully in the not too distant future, face to face as well. So with that, I think we'll close the session today. Many thanks.